In the 1840s, a British naturalist named Edward Forbes dragged a net through the Mediterranean Sea and came to a conclusion that would shape how we understood our planet for the next century. He declared, based on what he pulled up, that life simply could not exist in the deep ocean. Below 300 fathoms, roughly 550 meters, there was nothing. Just water, pressure, and darkness. He called it the Azoic Zone, the lifeless zone, and for decades the scientific world agreed with him. But here is the thing about Edward Forbes. He used the wrong tools in the wrong ocean and ignored the evidence that already told him he was wrong. His dredges were too small to catch the organisms that lived at those depths. The Aegean Sea, where he did most of his sampling, is naturally low in deep-sea biomass compared to other oceans. It was like failing to find lions in Canada and concluding that lions do not exist. And yet his idea stuck. It became the default assumption. The deep ocean was dead, and nobody questioned it. Nearly 200 years later, we are still making the same mistake. Not with the same words, obviously. Nobody publishes a paper today saying the deep ocean is lifeless. But the underlying assumption, the feeling that the deep sea is empty, barren, irrelevant, that still lingers. It lingers in how little money we spend exploring it. It lingers in how casually we talk about mining it. And it lingers in the surprise we express every single time a camera comes back with footage of something we never expected to find. We have visually observed less than 1,000th of 1% of the deep sea floor. That is a real number, published in Science Advances in 2024. The total area of deep ocean floor that human eyes have ever seen, through submersible windows or robot cameras, adds up to roughly 1/10 the size of Belgium. That is it. Everything we think we know about the largest habitat on Earth is based on that tiny sliver. And in just the last few years, that sliver has delivered discoveries so strange, so fundamentally unexpected, that they are rewriting the rules of biology, chemistry, and maybe even the origin of life itself. So tonight, we are going to go deep. We are going to look at the creatures we said could not exist, the chemistry we said was impossible, and the ecosystems we nearly destroyed before we even knew they were there, because it turns out the deep ocean has been trying to tell us something for a very long time, and we have been too busy looking up at the stars to listen. Let us start at the beginning, with the man who got it wrong. Edward Forbes was not a fool. He was actually one of the most respected naturalists of his time, a professor at the University of Edinburgh and a Fellow of the Royal Society. When he sailed aboard HMS Beacon in the early 1840s, surveying the marine life of the Aegean and Eastern Mediterranean, he was doing legitimate science. He lowered his dredges deeper and deeper, cataloguing what he pulled up, and what he noticed was a pattern. The deeper he went, the less life he found. In shallow waters his nets came up full. At moderate depths there was less, and at the deepest points he sampled there was almost nothing. So he drew a line. He extrapolated from his data and proposed that somewhere around 300 fathoms life simply stopped. Below that point was the Azoic Zone, a vast, dark, dead world where the pressure was too great and the resources too scarce for anything to survive. It was a reasonable hypothesis based on limited data. But there were two problems Forbes apparently chose to overlook. First, the Aegean Sea is not representative of the world's oceans. It is semi-enclosed, relatively shallow, compared to the open Atlantic or Pacific, and has naturally lower deep-sea biomass. Sampling the Aegean and drawing conclusions about the entire planet's deep ocean is like studying a parking lot and declaring that Earth has no forests. Second, and this is the part that really stings, there was already evidence that he was wrong. Other naturalists had reported pulling organisms from depths well below Forbes' supposed dead zone. Specimens of deep water corals and starfish had been collected from hundreds of fathoms before Forbes ever set sail. He either dismissed them or did not engage with them at all, and yet the Azoic hypothesis caught on. It became one of those ideas that everyone assumed was true because someone credible had said it once, and nobody bothered to really challenge it for years. It was comfortable, it fit a narrative. The deep ocean was dark and hostile, therefore nothing could live there. Case closed. But the ocean does not care about our cases, and in 1872, a ship set sail that would blow the Azoic hypothesis apart. The HMS Challenger left Portsmouth, England in December 1872 on what would become one of the most important scientific voyages in the human history. Over 3.5 years it travelled nearly 70,000 nautical miles, circling the globe and stopping to sample the ocean at 362 stations. The crew lowered nets, dredges and thermometers into the deepest water they could reach, and what came back up in those nets changed everything. Life. Lots of it. From thousands of metres down, in water that Forbes had declared sterile, the Challenger pulled up specimens of creatures that nobody had ever seen before. Strange fish, bizarre invertebrates, organisms adapted to total darkness and crushing pressure. Over the course of the expedition, the ship's scientists catalogued roughly 4,700 new species. The Azoic zone did not exist. It never had. The Challenger expedition is rightly celebrated as the birth of modern oceanography. Its 50 volumes of reports, published over nearly 2 decades after the voyage, fundamentally reshaped our understanding of the ocean. But here is what is less often discussed. The public did not really catch up. The scientific community moved on from the Azoic hypothesis, sure, but the cultural idea that the deep ocean is a dead, empty wasteland persisted for much longer than it should have. Part of this is because the deep sea is hard to visit. You cannot take a tourist submarine to the abyssal plain the way you can drive to a national park. Most people will never see a hydrothermal vent or a bioluminescent fish in person, and what you cannot see you tend to forget about. Part of it is also cultural. The deep ocean has always been associated with fear, with the unknown, with the underworld. The word abyssal comes from the Greek for bottomless. The Vidal Zone, the very deepest ocean trenches, is named after Hades. We talk about the deep as if it is a void, a gap, an absence, rather than an ecosystem. Jules Verne did not help. His 1870 novel, 20,000 Leagues Under the Sea, was a masterpiece of fiction, but it popularized the idea that water pressure would suspend objects at specific depths, that sunken ships would float in mid-water, and that the deep sea was a kind of watery graveyard. The concept had already been debunked in scientific circles, but Verne's version lodged itself in public imagination. To this day, no mention of the Mariana Trench is complete without the cliché that it is so deep Mount Everest could disappear inside it, which is technically true, but also a perfect example of how we describe the deep ocean, by comparing it to things we already know, rather than on its own terms. Here is the reality. The deep ocean, defined as everything below 200 meters, makes up over 90% of the biosphere. It covers 66% of the entire planet. It is the single largest habitat on Earth, and it is teeming with life. Not just microbes clinging to existence, though there are plenty of those. Complex ecosystems with food chains, predator-prey relationships, symbiotic partnerships, and biodiversity that rivals anything on land. And we have barely looked at it. The Science Advances paper from 2024 laid out the numbers in stark detail. 65% of all in situ. Visual observations of the deep sea floor in their dataset were conducted within 200 nautical miles of just 3 countries, the United States, Japan and New Zealand. 97% of all deep-sea dives ever compiled were conducted by just 5 countries. The remaining 190-odd nations on Earth have contributed almost nothing to the visual record of the deep ocean. And even the countries that have explored the most have barely scratched the surface, or rather, barely scratched the bottom. Governments allocate, on average, only 1.7% of their annual research budgets to exploring beneath the ocean surface. More people have travelled to space than have travelled to the deep ocean floor. We have better maps of Mars than we do of our own seabed. So when scientists tell you that every deep-sea expedition turns up new species, they are not exaggerating. It is common for 50% of all organisms collected on a single expedition to be new to science. On some voyages, that number climbs past 90%. We are not at the stage of finding the last few undiscovered species in a well-catalogued environment. We are at the stage of opening a door to a room we did not know existed and finding it full of furniture. In March 2025, the Nippon Foundation and Nekton Ocean Census announced a staggering milestone. Since their collaborative program launched, they had identified 866 new marine species, not collected over decades, identified in a concentrated burst of targeted expeditions. Sharks, sea butterflies, mud dragons, bamboo coral, water bears, sponges, shrimp, crabs, and things that do not fit neatly into any category we have built. And those were just the ones they had managed to formally describe. The backlog of specimens waiting to be named and catalogued stretches back years, because the process of formally describing a new species is slow, painstaking work, and the ocean keeps delivering faster than taxonomists can keep up. We thought the deep was dead, then we found out it was alive, but we still assumed it was simple, sparse, barely hanging on. That assumption was wrong too, because the deep ocean was not just surviving, it was thriving in ways we could not have imagined, and the discovery that proved it changed biology forever. And one more thing to keep in mind as we go. The number 240,000, the total count of formally described marine species, is not a ceiling we are approaching. It is a floor we are barely standing on. Estimates of total marine species range from 1 million to over 2 million. Some marine taxonomists believe even those estimates are conservative, because the deep ocean, which comprises the vast majority of habitable space in the sea, has yielded such a tiny sample that our statistical models cannot account for what remains hidden. We are not completing a puzzle. We are still opening the box. In February 1977, a group of geologists climbed aboard the research vessel Canor and sailed to a place called the Galapagos Rift, about 400 miles off the coast of Ecuador. They were not looking for life. They were looking for heat. Specifically, they wanted to find hydrothermal vents, cracks in the ocean floor where superheated water from the Earth's interior seeps out into the sea. Geophysicists had predicted these vents should exist along mid-ocean ridges, the underwater mountain chains where tectonic plates are pulling apart, but nobody had ever seen one. There were no biologists on the expedition. Why would there be? The deep ocean, after all, was a desert. Sunlight cannot reach the seafloor at those depths, and without sunlight you cannot have photosynthesis. Without photosynthesis you cannot have plants. Without plants you cannot have a food chain. Without a food chain you cannot have complex life. That was the logic. It was clean, it was elegant, and it was completely wrong. When the scientists lowered their instruments and camera systems to the seafloor at the Galapagos Rift, roughly 2,500 meters below the surface, they found the vents they were looking for. Hot water was indeed streaming out of cracks in the volcanic rock, shimmering in the surrounding deep-sea cold, but clustered around those vents in thick, dense communities was something nobody had predicted. Life. Not just a few scattered organisms eking out a living, thriving, abundant, wildly diverse life. Giant tube worms, some of them over 1 meter long with blood-red plumes waving in the current. Enormous clams, swarms of strange shrimp, mats of white bacterial film covering the rocks. It was an oasis in the deep, a garden growing in a place where every rule of biology said a garden could not grow. Robert Ballard, the geologist and submersible expert who would later become famous for discovering the wreckage of the Titanic, was part of the team. Years later, he described the moment the images came back from the seafloor. Everyone sat around speechless, he said. It was like processing a non-linear equation. It was pretty amazing to find these creatures. The question, of course, was how. How were these animals surviving without sunlight? What were they eating? The tube worms had no mouth, no stomach, no gut. They were not grazing on anything. The shrimp were not hunting fish. The entire community seemed to be powered by something invisible. The answer came 4 years later, in 1981, from a Harvard graduate student named Colleen Cavanaugh. She proposed something radical. Bacteria, she argued, were living inside the bodies of the tube worms, packed into a specialized organ called the trophosome. These bacteria were not photosynthetic, they were chemosynthetic. Instead of using sunlight to convert carbon dioxide into food, they were using hydrogen sulfide, a chemical that pours out of hydrothermal vents, as their energy source. The bacteria were feeding the worms from the inside, and the worms were providing the bacteria with a safe home and a steady supply of the chemicals they needed. It was a symbiosis, a partnership, and it was running on the chemistry of the earth itself, not the light of the sun. The concept of chemosynthesis was not entirely new. A Russian microbiologist named Sergei Winogradsky had described the process back in the 1880s, studying sulfur-oxidizing bacteria in hot springs on land. But nobody had imagined that chemosynthesis could power entire ecosystems of large, complex animals. Nobody had imagined that you could have a food chain rooted, not in sunlight, but in volcanic chemistry, hydrogen sulfide, methane, and the raw heat of the planet's interior. That single discovery, verified and confirmed over the following years, rewrote one of the most fundamental assumptions in biology. Before 1977, every textbook in the world stated, explicitly or implicitly, that all complex life on Earth was ultimately dependent on photosynthesis. The sun was the engine. Everything else was downstream. The hydrothermal vent discovery proved that this was not true. Life could thrive completely independent of the sun, powered instead by the Earth's own internal chemistry. And the vents themselves turned out to be even more extreme than anyone initially realized. 2 years after the Galapagos discovery, scientists exploring another section of the mid-ocean ridge found a different kind of vent. These were not gentle seeps of warm water. These were violent eruptions, superheated fluid, reaching temperatures of 400 degrees Celsius, hot enough to melt lead, blasting out of tall chimney-like structures on the seafloor. The fluid was black with dissolved minerals, iron, copper, zinc, manganese, precipitating out of solution the instant it hit the frigid surrounding water. They called them black smokers, and animals were living right next to them. Not meters away, right there on the chimney walls, inches from water that would cook them alive if they drifted too close. The temperature gradient around a black smoker is one of the steepest on Earth. You can go from 400 degrees to 2 degrees in the space of a few centimeters. The organisms that colonize these chimneys live in a razor-thin habitable zone, balancing between being boiled and being frozen. One of the most remarkable examples was discovered later in Antarctic waters. The Yeti crab, Kiwa tyleri, is a pale hairy crustacean that clusters around hydrothermal vents on the seafloor near Antarctica. Researchers have documented densities exceeding 700 individual crabs per square metre on vent chimneys. They are packed so tightly that they look like a living carpet, and they have almost no room for error. Stray too far from the vent and the water temperature drops to near freezing, cold enough to kill them. Get too close and they cook. Their entire existence is confined to a band of survivable temperature that is sometimes only centimetres wide. The Yeti crabs farm bacteria on their hairy arms. They wave their arms through the chemical-rich vent water, letting bacteria grow on the setae, the tiny hair-like structures, and then they eat the bacteria. They are farmers in the abyss, cultivating their own food supply in one of the most hostile environments on the planet. But hydrothermal vents were just the beginning. Over the following decades, scientists discovered that chemosynthesis was not limited to volcanic hotspots on mid-ocean ridges. It was happening in other places too. Cold seeps where methane and hydrogen sulfide bubble up slowly through the seafloor sediment support their own communities of specialized organisms. Mussels, clams, and tube worms cluster around these seeps, hosting chemosynthetic bacteria in their tissues, living on chemistry rather than sunlight, just like their cousins at the hot vents. Then there are whale falls. When a whale dies and its carcass sinks to the deep ocean floor, it creates a temporary ecosystem that can persist for decades. First, the large scavengers arrive, hagfish and sleeper sharks, stripping the flesh. Then smaller organisms move in on the remaining tissue. And finally, as the whale's bones begin to decompose, releasing sulfides, a chemosynthetic community establishes itself on and around the skeleton. Bone-eating worms called osedacs bore into the whale's remains, consuming the lipids inside. Bacteria colonize the bones, using the sulfide to fuel chemosynthesis. A dead whale on the seafloor becomes an island of life in the deep desert, a self-contained ecosystem that runs for 50 to 100 years. Woodfalls work similarly. When a tree trunk or large piece of wood sinks to the abyss, specialized organisms colonize it. Some of the most recently described deep-sea species are mollusks that live exclusively on sunken wood, feeding on the organic material or hosting bacteria that break it down. One species of Chiton ferrariella populi was discovered in 2024 in the Izu Ogasawara Trench at a depth of 5,500 meters, living on a piece of wood that had drifted down from the surface world. It was formally named in 2026 after a public naming competition, making it one of the first deep-sea species to receive its name from the thousands of ordinary people voting online. The discovery of chemosynthetic ecosystems did more than just add a new chapter to biology textbooks. It fundamentally changed how we think about where life can exist, not just on Earth but beyond it. Jupiter's moon Europa has a subsurface ocean of liquid water beneath a shell of ice. Saturn's moon Enceladus has one too, and the Cassini spacecraft actually flew through geysers erupting from its south pole, detecting salt, organic molecules, and the chemical ingredients for life. Neither of these moons receives enough sunlight to drive photosynthesis in their oceans. But if hydrothermal vents exist on their sea floors, and there is strong theoretical reason to believe they might, then chemosynthesis could potentially fuel life there, just as it does in the deepest parts of our own ocean. Before 1977, the search for extraterrestrial life was almost entirely focused on finding conditions suitable for photosynthesis. A planet needed to be in the habitable zone, the right distance from its star for liquid water to exist on the surface bathed in sunlight. After the vent discovery, that search expanded dramatically. Suddenly, any world with liquid water, chemical energy, and a heat source became a candidate. The discovery of life at hydrothermal vents did not just change marine biology, it redirected the entire field of astrobiology. And all of this, every single one of these discoveries, came from a place that we once declared was dead. The deep ocean floor, the Azoic zone, the void below 300 fathoms. Forbes said nothing lived there. The vent said everything could live there if you gave it the right chemistry. But we were not done being wrong, because even after we accepted that life could survive without sunlight, even after we mapped hundreds of hydrothermal vents and cold seeps, we still believed we understood the limits, the pressure, the cold, the darkness. There was a flaw, we said, a point below which biology simply could not function. Fish could not survive below a certain depth. The physics of their cells would not allow it. That turned out to be wrong too, and the creature that proved it was not some terrifying deep-sea monster. It was a small, translucent, tadpole-shaped fish that most people have never heard of. The tube worm, Riftia pachyptila, deserves a moment of its own because it is one of the strangest organisms ever found on Earth. It grows to over 2 meters in length, anchored to the rock near a hydrothermal vent. It has a brilliant crimson plume that waves in the current, packed with blood vessels carrying hemoglobin, the same iron-based molecule that makes our blood red. It uses that hemoglobin to absorb hydrogen sulfide from the vent water and oxygen from the surrounding sea water simultaneously, then delivers both chemicals to the bacteria living inside its body. Here is the part that matters. The tube worm has no mouth. It has no stomach. It has no digestive tract of any kind. It cannot eat. It does not eat. It relies entirely on its internal bacteria to convert hydrogen sulfide and carbon dioxide into organic molecules, sugars and fats that nourish the worm. The relationship is so complete, so absolute, that the worm has evolutionarily given up its ability to feed itself. It is a living housing unit for bacteria, a biological apartment building whose sole function is to provide shelter and raw materials in exchange for food it cannot make on its own. And it grows fast. In environments where food falls from the surface at a rate measured in milligrams per square metre per day, the tube worm, powered by chemosynthesis, grows at rates comparable to organisms in the sunlit tropics. Some studies have measured growth rates of over 80 centimetres per year. In the deep sea desert, these vents are not just oases, they are the most productive ecosystems per unit area on the planet. But the vents are also temporary. A hydrothermal vent system driven by volcanic activity typically lasts a few decades, maybe a century if conditions are right. The magma shifts, the plumbing of the crust changes, and the vent dies. When that happens, the community dies with it. The tube worms, the clams, the shrimp, all of them are gone within months or years. And then, somewhere nearby, a new vent opens, and the colonization process starts over. How organisms find these new vents, how their larvae disperse through miles of lightless ocean to settle on a crack in the rock that did not exist a few years ago, is one of the biggest unsolved questions in deep-sea ecology. The answer may involve something remarkable. Some vent organisms appear to use whale falls and wood falls as stepping stones. When a dead whale sinks to the ocean floor and its bones begin producing hydrogen sulfide, chemosynthetic organisms colonize it. These whale fall communities have been found to include species that are closely related to vent species, suggesting that dead whales serve as temporary islands of suitable habitat scattered across the abyssal plain. A whale carcass might sustain a chemosynthetic community for 50 to 100 years, long enough for larvae to hatch, disperse, and potentially reach the next vent or the next whale fall. The deep ocean's food chain is connected in ways that we are only beginning to map. Cold seeps add another dimension to this picture. Unlike hydrothermal vents, which are driven by volcanic heat and tend to be geologically young and short-lived, cold seeps are places where methane and hydrogen sulfide ooze slowly through the sediment, often along fault lines or at the edges of continental shelves. The fluid is not hot. It is often at ambient, deep-sea temperature, near freezing. But the chemistry is there, and where there is chemistry, there is chemosynthesis, and where there is chemosynthesis, there is life. Cold seep communities can persist for thousands of years, far longer than vent communities. They support their own assemblages of specialized organisms, including mussels and clams that host chemosynthetic bacteria in their gills, ice worms that live on methane hydrate deposits, and vast bacterial mats that cover the sediment in white or orange films. Some cold seep sites also feature natural deposits of methane hydrate, a crystalline solid that forms when methane gas is trapped in a cage of water molecules under high pressure and low temperature. Methane hydrate looks like ice, but burns when you light it, because it is releasing trapped methane. These deposits are widespread on continental margins around the world and contain an estimated amount of carbon that dwarfs all other fossil fuel reserves on Earth combined. The organisms living at cold seeps interact with these methane deposits in complex ways. Certain archaea, working in partnership with bacteria, consume methane before it can escape into the water column. This microbial methane consumption is an ecosystem service of global significance, because methane is an extremely potent greenhouse gas. The deep-sea microbes that eat methane at cold seeps are, in a very real sense, part of the planet's climate regulation system. They are a brake on warming, operating silently in the dark, entirely without our knowledge or assistance. And then there is bioluminescence, the production of light by living organisms, which turns out to be one of the most underappreciated phenomena in the deep sea. Up to 90% of all organisms in the deep ocean can produce light. This is not a rare trick or an oddity. It is the dominant mode of communication, hunting and defense below 200 meters. The reason is simple. Below the sunlit zone, the ocean is dark, permanently, absolutely dark. In an environment where no ambient light exists, any organism that can produce its own light has a massive advantage. Bioluminescence serves different purposes for different species. Some use it to attract prey, like the anglerfish, with its glowing lure dangling in front of its mouth. Some use it to communicate with potential mates, flashing specific patterns in a language of light that scientists are only beginning to decode. Some use it for camouflage, a strategy called counterillumination, in which an animal produces light on its underside that matches the faint glow filtering down from the surface, eliminating its silhouette and making it invisible to predators looking up from below. And some use it as a weapon. The deep-sea hooked squid, Taningia danae, is one of the largest squid in the deep ocean. It has two enormous photophores, light-producing organs, at the tips of two of its arms. These are the largest bioluminescent organs known in any animal on Earth. In 2024, researchers from the University of Western Australia filmed a Taningia danae attacking their camera at about 1,000 meters depth in the Pacific. The squid descended on the camera, apparently mistaking it for prey, and just before striking it flashed its photophores in a blinding burst of light. The flash is thought to stun prey, like a biological flashbang grenade. The squid then wrapped its arms around the camera, engulfing it completely, before releasing and vanishing back into the dark. This species was first observed alive only about 19 years ago. Before that, everything we knew about Taningia danae came from dead specimens found in the stomachs of whales or caught accidentally in deep water trawls. Every living encounter since has added something new to our understanding. And every encounter has happened by chance, because we do not yet have the technology to reliably track and follow these animals in their own environment. The deep ocean is a world of light in the dark, where organisms have evolved a chemical vocabulary of flashes, glows and pulses that serves every purpose, from romance to warfare. And we have been studying it for less than 50 years. The Hadal Zone is the deepest layer of the ocean, named after Hades, the Greek god of the underworld. It begins at roughly 6,000 meters and extends all the way down to nearly 11,000 meters at the bottom of the Mariana Trench. It exists almost exclusively in long, narrow, V-shaped trenches, the places where one tectonic plate dives beneath another, dragging the seafloor down into the Earth's mantle. There are 46 individual Hadal habitats worldwide, and together they account for less than 0.25% of the entire ocean floor, but they make up over 40% of the ocean's total depth range. These are places of absolute darkness. No photon of sunlight has ever reached the bottom of the Mariana Trench. The temperature hovers just above freezing, between 1 and 4 degrees Celsius, and the pressure is beyond anything we encounter in daily life. At the bottom of the Challenger Deep, the deepest known point in the ocean, at 10,911 meters, the water pressure exceeds 1,100 atmospheres. That is roughly 16,000 pounds per square inch. For context, the pressure at sea level is about 14.7 pounds per square inch. The deep ocean presses on you with a force that would instantly crush a submarine not specifically engineered to withstand it. In 2014, an advanced robotic vehicle called Nereus, built by the Woods Hole Oceanographic Institution, imploded at a depth of 9,990 meters in the Kermadec Trench, just snapped like a tin can. For a long time, the prevailing assumption was that very little could survive at those depths. Life decreases with depth. That much is true. Both the abundance and the biomass of organisms decline as you descend. The Hadal Zone is not teeming with life the way a coral reef is. but the assumption went further than that. Scientists believed there was a hard limit, a physiological ceiling beyond which fish simply could not function. The reasoning was elegant and well supported by chemistry. Fish, like all animals, need their proteins to fold correctly to work. At extreme pressures, proteins tend to unfold, to denature, the same way an egg whites turn solid when you heat it. To counteract this, deep-sea fish accumulate a compound called trimethylamine N-oxide, or TMAO. It is an osmolyte, a molecule that stabilizes proteins under pressure. The deeper a fish lives, the more TMAO it needs in its cells, but TMAO has a limit. At some point you simply cannot pack any more of it into your cells without disrupting other biological processes. The fish becomes hyperosmotic, meaning its internal fluid concentration overwhelms its ability to regulate. Based on the observed relationship between depth and TMAO concentration, scientists calculated that the theoretical maximum depth for fish was somewhere around 8,200 to 8,500 meters. Below that, the chemistry of life in a fish's body should stop working. Proteins would unravel, enzymes would fail. The fish would, in a very real sense, dissolve from the inside. For years this prediction seemed solid. Researchers had sent baited cameras to the deepest parts of 14 different ocean trenches. None of them had ever captured footage of a fish below 8,200 meters. Trawling expeditions by Danish and Soviet researchers had dropped nets 134 times to depths greater than 6,000 meters. No fish ever came up from a tow below the theoretical limit. The math seemed right. The observations confirmed it. The deepest quarter of the ocean was apparently fishless. But then, in 2017, a team led by marine scientist Alan Jameson from the University of Western Australia lowered a baited camera into the Mariana Trench and filmed a snailfish at 8,178 meters. It was pale, translucent, and shaped roughly like a tadpole. It was hovering just above the sediment, feeding on tiny crustaceans attracted by the bait. It was deeper than any fish had been reliably recorded before, and it was clearly alive and functioning, not struggling, not dying. Just going about its business in the most extreme environment a vertebrate had ever been observed in. 5 years later, in 2022, the same team sent cameras down again, this time into the Izu Ogasawara Trench south of Japan, and they found another snailfish. This one was at 8,336 meters, 158 meters deeper than the Mariana record, sitting right at the ragged edge of the supposed impossible. The fish was a juvenile, small and delicate. Its species has been identified as belonging to the genus Pseudoliparis, but it has not been formally named yet. It was attracted to the camera by the bait, and it appeared healthy, active, and perfectly adapted to its environment. Jameson described the discovery carefully. "They can't really go any deeper," he said. This is essentially the limit of what fish can do. But the fact that they were there at all, at a depth scientists had calculated should be uninhabitable, demanded explanation. And when researchers examined snailfish specimens in the lab, they found a suite of adaptations that were as surprising as the depth record itself. Start with the skull. In most fish, the skull is a rigid box of bone protecting the brain. In the deepest snailfish, the skull has gaps in it, incomplete ossification. The bones are not fully formed. This sounds like a defect, but it is actually an adaptation. At extreme pressure, a rigid skull would create a pressure differential between the inside and outside of the head. By leaving gaps, the fish allows pressure to equalize across the skull, preventing the kind of catastrophic structural failure that destroyed the Nereus robot. Their skin is transparent and lacks pigment. There is no point investing energy in coloration when you live in total darkness. Their muscles are thinner than those of shallow water relatives. Their organs and eggs are enlarged. Their eyes are reduced to the point of being functionally useless, and genetic analysis has confirmed that they lack several key photoreceptor genes. They cannot see. They have evolved past the need for sight. And then there is the TMAO. The deepest snailfish have TMAO concentrations that push right up against the theoretical maximum. They are living at the chemical limit. But the Izu Ogasawara Trench has one small advantage over the Mariana. It is slightly warmer, by about a fraction of a degree, and TMAO is slightly more effective at higher temperatures. That tiny difference in water temperature, a difference that you or I would never notice, appears to be the margin that allows a fish to survive 158 meters deeper than anyone previously thought possible. Each ocean trench has its own species of snailfish. They have evolved independently in these isolated deep water habitats, adapting to the specific conditions of their particular trench. And because they cannot decompress to migrate between trenches, the distances between them too vast and the shallow water above too inhospitable, they are effectively prisoners of their depth. Each species is endemic, found nowhere else on Earth. The Mariana snailfish, Pseudoliparis swire, was formally described in 2018. It reaches about 28 centimeters in length and feeds on tiny amphipods and crustaceans. It is somewhat remarkably the top predator in its environment, not because it is particularly fearsome, but because there is almost nothing else down there that eats what it eats. At those depths the food chain is short. Organic matter drifts down from the surface in a slow, continuous rain called marine snow. Tiny crustaceans and amphipods feed on this material, and the snailfish feeds on the crustaceans. That is the entire food web. Simple, fragile and utterly dependent on what falls from above. But the snailfish are not the only inhabitants of the Hadal Zone. In 2021, Chinese researchers aboard the deep-sea submersible Fenduje conducted 33 dives into the Mariana Trench, reaching depths between 6,000 and 10,900 meters. Over the course of the expedition, they collected thousands of sediment and water samples, and when they analysed what they found, the results were astonishing. Over 7,000 species of microbes were identified, and roughly 90% of them had never been documented before. These were not just a few hardy survivors clinging to existence in the deep. This was a diverse, complex microbial community with its own ecological dynamics, its own food webs, its own relationships. The researchers also collected specimens of the Hadal amphipod Hirundellea gigas, a tiny crustacean that somehow manages to move between different deep-sea trenches. How it does this, given the crushing pressure changes involved, is still poorly understood. They also encountered the snailfish Pseudoliparis swirei, confirming its status as the deepest living fish. The findings were published in Cell, one of the world's most prestigious scientific journals, and they painted a picture of the Hadal Zone that was far richer and more complex than anyone had anticipated. We assumed the deepest ocean was too hostile for complex life. We calculated the theoretical limits and drew a line, and then a small, translucent tadpole-shaped fish swam across that line and kept going. But the deep ocean was not done in surprising us, because if the snailfish showed that life could push further than we thought, the next discovery suggested that the deep ocean was not just supporting life. It might be producing the one thing that all complex life needs to survive, and it was doing it in a way that nobody had ever imagined was possible. The Hadal Zone is not just about fish. It is about an entire suite of organisms that have evolved to thrive in conditions that would destroy almost anything from the surface world. Amphipods, for example, are small crustaceans that are found everywhere in the ocean, from the tide pools to the deepest trenches. But in the Hadal Zone, they undergo something extraordinary. Some species exhibit gigantism, growing far larger than their shallow water cousins. The supergiant amphipod Allicella gigantea can reach 34 centimeters in length, roughly the size of a rabbit. In a world where food is scarce and arrives in unpredictable pulses, being big has advantages. You can store more energy. You can eat larger pieces of whatever falls from above. You can survive longer between meals. These amphipods are scavengers. They feed on marine snow and on the carcasses of larger animals that sink to the trench floor. When researchers lower baited cameras into Hadal trenches, the amphipods are usually the first things to appear, swarming the bait in dense writhing clouds. They can detect food from remarkable distances, navigating by chemical cues in the water column, and they arrive fast. Within hours of deploying a baited lander at 10,000 meters, you can have thousands of amphipods competing for the food. The deep sea may be quiet, but it is not slow. Sea cucumbers are another Hadal success story. These soft-bodied echinoderms, relatives of starfish and sea urchins, have adapted to life in the deepest trenches with a grace that their appearance might not suggest. At Hadal depths, sea cucumbers are often the most abundant large organisms on the seafloor. They creep along the sediment, vacuuming up the thin layer of organic material that settles from above. Some species have become so specialized for this lifestyle that they are essentially living conveyor belts, ingesting sediment at one end and expelling it at the other, extracting every last calorie of nutrition from the mud. In the Mariana Trench, researchers have found sea cucumbers at depths exceeding 10,000 meters, making them among the deepest multicellular organisms ever observed. One species, documented during the Chinese Fendouzha expeditions, was filmed gliding across the sediment at the very bottom of the Challenger Deep. It was pale, almost translucent, and moved with a slow, deliberate rhythm that seemed almost meditative. There is something both humbling and oddly comforting about knowing that at the deepest point on Earth, in absolute darkness, under pressure that would crush a nuclear submarine, a sea cucumber is going about its day. Foraminifera are another group that reaches extraordinary proportions in the deep. These single-celled organisms build elaborate shells or tests from minerals they extract from the water. At Hadal depths, some Foraminiferae species grow to several centimeters, enormous for a single cell. Their tests accumulate on the seafloor over millions of years, forming thick layers of calcareous ooze that eventually lithify into rock. The White Cliffs of Dover, those iconic chalk formations in England, are made largely of ancient foraminifera shells. The deep ocean is not just a habitat, it is a geological engine, turning biology into stone over timescales that dwarf human civilization, and it is this geological timescale that makes the mining question so urgent. When we talk about destroying deep-sea habitat, we are not talking about damage that can be repaired in a human lifetime, or even in the lifetime of a civilization. Polymetallic nodules grow at a rate of roughly 1 to 10 millimeters per million years. To form a single nodule the size of a potato takes somewhere between 3 and 10 million years. The habitat they provide, the hard substrate on which sessile organisms can anchor, the electrochemical environment that may produce dark oxygen, the microhabitats in the crevices between clustered nodules, all of this takes geological time to develop. When a collector vehicle scrapes a section of the Clarion-Clipperton zone clean, the nodules are gone. The organisms that lived on them are gone. The microbial communities that inhabited the sediment between them are disrupted. And the clock to replacement starts at zero, measured not in years or decades, but in millions of years, by any standard that matters to human decision-making. The destruction is permanent. The sediment plumes created by mining add another layer of damage. The CCZ seafloor is covered with a fine layer of sediment that has settled over millennia, undisturbed by currents or waves. When a mining vehicle churns through this sediment, it kicks up clouds of fine particles that can travel enormous distances. Studies using current modelling have shown that these plumes can spread for tens of kilometres from the mining site before settling. When they settle, they blanket the seafloor in a layer of redeposited sediment that smothers organisms, clogs respiratory structures and changes the chemical composition of the surface layer. For filter feeders, organisms that strain food particles from the water, a sediment plume is catastrophic. It is the equivalent of a dust storm that never ends, filling their feeding apparatus with inedible grit. For organisms that rely on chemical cues to find food or mates, the disturbance in water chemistry caused by resuspended sediment can disrupt communication and navigation. And for organisms that have evolved over millions of years in an environment of absolute stillness, any physical disturbance is an event for which they have no evolutionary preparation. The International Seabed Authority has been negotiating the mining code that would govern commercial extraction in the CCZ since the 1990s. As of 2026, the code is still not finalised. Environmental organisations, a growing number of governments, and many deep-sea scientists have called for a moratorium on mining until we have a better understanding of what we would be destroying. Others, particularly mining companies and some resource-dependent nations, argue that the metals in polymetallic nodules are essential for the green energy transition and that delays in mining are delays in decarbonisation. The argument is not simple. Both sides have legitimate points, but the fundamental asymmetry remains. We know a great deal about the economic value of the metals in polymetallic nodules. We know almost nothing about the ecological value of the ecosystems those nodules support. We are being asked to make an irreversible decision on the basis of incomplete information, and the history of deep-sea science teaches us, over and over again, that every time we assume the deep ocean is less important, less complex, or less alive than it appears, we turn out to be wrong. Oxygen is the one thing that all complex animal life needs. Without it, there are no fish, no crabs, no worms, no squids. Bacteria and archaea can get by without it, running their metabolisms on sulfur or methane or iron. But animals, creatures with tissues and muscles and nervous systems, need oxygen to power their cells. And for the entirety of human scientific history, we have believed that oxygen comes from one source and one source only: photosynthesis. Plants on land and phytoplankton in the ocean use sunlight to split water molecules, releasing oxygen as a byproduct. Roughly half the oxygen we breathe is produced by microscopic organisms floating near the surface of the sea, bathing in sunlight. The other half comes from terrestrial plants and forests. That is the equation. Sun plus water plus biology equals oxygen. No sun, no oxygen. The deep ocean, where sunlight never reaches, was therefore understood to be an oxygen consumer, not an oxygen producer. Whatever dissolved oxygen existed in deep water had been carried there by ocean currents, cold surface water, sinking at the poles and flowing along the seafloor in a global conveyor belt that takes centuries to complete its circuit. The creatures living in the deep use that oxygen for respiration, slowly depleting it. Every experiment ever conducted on the deep sea floor had confirmed this. You put a chamber on the bottom of the ocean, seal it, and measure the oxygen inside. It goes down, always down. Organisms are breathing, consuming, depleting. That is how the deep sea works, until it wasn't. Andrew Sweetman is a marine scientist at the Scottish Association for Marine Science in Scotland. For over a decade, he's been studying the biology of the deep sea floor in the Clarion Clipperton Zone, a vast region of the Pacific Ocean stretching nearly 4,500 miles between Hawaii and Mexico. The CCZ, as it is known, is one of the most intensely studied areas of the deep ocean, not because of its biology, but because of its geology. The seafloor there is covered with polymetallic nodules, potato-sized lumps of mineral-rich rock containing manganese, nickel, cobalt, copper, and lithium. These are the metals that go into batteries, and the mining industry wants them badly. Sweetman's job was to measure how fast organisms on the seafloor consumed oxygen, a standard metric for understanding the health and activity of deep-sea communities. He used benthic chamber landers, devices that descend to the seafloor, land on the sediment, and seal off a small patch of ocean bottom inside a closed chamber. Sensors inside the chamber measure how oxygen levels change over time. In a healthy deep-sea community, the oxygen should slowly decrease as organisms respire. It is a routine experiment. done hundreds of times by dozens of research groups around the world. But when Sweetman got his data back from the CCZ, something was wrong. The oxygen was not going down, it was going up. More oxygen was accumulating inside the sealed chambers than was being consumed. In some experiments, oxygen levels tripled over a 2-day period. That is not a subtle anomaly. That is the doubter screaming that something unexpected is happening. Sweetman's first instinct, understandably, was that his instruments were broken. He took the sensors home, recalibrated them and sent them back down. Same result. He tried different sensors. Same result. He tried a completely independent measurement technique, the Winkler method, a classical chemical procedure for measuring dissolved oxygen that has nothing to do with electronic sensors. Same result. Over the course of 10 years across multiple expeditions, the anomalous readings kept appearing. When we first got this data, we thought the sensors were faulty, Sweetman later said, because every study ever done in the deep sea has only seen oxygen being consumed rather than produced. We would come home and recalibrate the sensors, but over the course of 10 years, these strange oxygen readings kept showing up. Eventually, he became convinced the signal was real, and when he started looking for a cause, one element kept appearing in the experiments that showed oxygen production: polymetallic nodules. The chambers that contained nodules on the seafloor showed the oxygen increase. Chambers placed on bare sediment without nodules did not. The nodules were doing something. In the summer of 2023, Sweetman contacted Franz Geiger, a professor of chemistry at Northwestern University, who had previously studied the electrochemical properties of rust and salt water. Together, they developed a hypothesis that sounded almost too strange to be real. Polymetallic nodules are made of layers of different metals, primarily manganese oxide, with pockets of nickel, cobalt, copper, and iron. These metals have different electrochemical properties. When stacked together in layers, with salt water flowing between them, they can create a separation of electrical charges. The same basic principle that makes a battery work. The nodules, sitting on the dark sea floor for millions of years, were acting as natural geobatteries. To test this, Sweetman shipped several pounds of nodules from the Pacific seafloor to Geiger's laboratory at Northwestern. The team measured the electrical voltage on the surface of individual nodules and found readings of up to 0.95 volts. That is a remarkable number, because just 1.5 volts, the voltage of a standard AA battery, is enough to split a water molecule into hydrogen and oxygen through a process called seawater electrolysis. A single nodule could not quite get there on its own, but polymetallic nodules do not sit alone on the seafloor. They cluster together, sometimes densely, and when batteries are connected in series, their voltages add up. It appears that we discovered a natural geobattery, Geiger said when the results were published. These geobatteries are the basis for a possible explanation of the ocean's dark oxygen production. The study was published in Nature Geoscience on July 22, 2024, and it landed like a bomb in the scientific community. The researchers had named the phenomenon dark oxygen, oxygen produced in complete darkness, without any involvement of photosynthesis or sunlight. The implications were staggering. If rocks on the ocean floor could produce oxygen through electrochemistry, then the story of oxygen on Earth might be more complicated than we thought. For aerobic life to begin on the planet, there had to be oxygen, Sweetman said after the publication, and our understanding has been that Earth's oxygen supply began with photosynthetic organisms. But we now know that there is oxygen produced in the deep sea where there is no light. I think we therefore need to revisit questions like where could aerobic life have begun. Jeff Marlowe, an assistant professor of biology at Boston University and a co-author on the paper, went even further. The fact that oxygen can be made independently of the surface world has a lot of exciting implications for the distribution of animal life, not only in Earth's oceans, but also potentially beyond our planet, he said. If oxygen production does not require sunlight, then the moons of Jupiter and Saturn with their subsurface oceans and mineral-rich seafloors, become even more plausible candidates for complex life. But the dark oxygen discovery also ignited a fierce debate that is still ongoing. As of 2025, no other research group has independently replicated Sweetman's findings, and in November 2025, a team of scientists published a detailed critique in Frontiers in Marine Science, arguing that the methodology had significant problems. They pointed out that numerous prior studies using functionally similar equipment in the same region, the Clarion-Clipperton Zone, had measured only oxygen consumption, not production. The critics raised questions about whether the experimental setup might have disturbed the seafloor sediment in a way that exposed reactive nodule surfaces that would normally be buried, creating a transient electrochemical effect rather than an ongoing natural process. The debate is important, and it is exactly how science is supposed to work. Extraordinary claims require extraordinary evidence, the critics wrote, echoing the old Carl Sagan maxim. And they are right. If dark oxygen is real, it is one of the most significant discoveries in Earth science in decades. If it is an artifact of the experimental method, it is a cautionary tale about how hard it is to study the deep ocean without disturbing it. But the timing of the discovery added a layer of urgency that went far beyond academic debate. The paper was published in July 2024, right in the middle of active international negotiations at the International Seabed Authority in Kingston, Jamaica, where diplomats and mining industry representatives were arguing over whether and how to allow commercial deep-sea mining. The dark oxygen findings were cited at the negotiating table within days of publication. If these nodules are producing oxygen that deep-sea ecosystems depend on, then removing them by the millions of tons could destroy an ecosystem function we only just discovered. 16 companies currently hold exploration contracts in the Clarion-Clipperton Zone. The polymetallic nodules they want to extract are the same objects that might be producing dark oxygen. Franz Geiger put it bluntly. The polymetallic nodules that produce this oxygen contain metals such as cobalt, nickel, copper, lithium, and manganese, which are all critical elements used in batteries. Several large-scale mining companies now aim to extract these precious elements from the seafloor at depths of 10,000 to 20,000 feet below the surface. We need to rethink how to mine these materials so that we do not deplete the oxygen source for deep-sea life. Whether dark oxygen turns out to be a fundamental new process or a fascinating anomaly that needs further investigation, one thing is clear. We thought we understood where oxygen comes from. We were so confident in that understanding that we built our entire model of planetary habitability around it. And then a marine scientist studying mud at the bottom of the Pacific found something that made us question the whole framework. But the nodules were not the only discovery in recent years that forced the deep-sea science community to reckon with how little it actually knows. Because while Sweetman was studying rocks on the seafloor, something appeared on camera that scientists had been trying to see for a century. Something that had eluded every expedition, every submarine, every remotely operated vehicle ever sent into the deep. And when we finally saw it, it did not look like a monster at all. The colossal squid was first described in 1925, not from a living animal swimming in the ocean, but from 2 tentacles pulled out of the stomach of a sperm whale that had been killed by commercial hunters in the Southern Ocean. A scientist named Robson examined the tentacles, noted the distinctive rotating hooks on their clubs, and concluded that they belonged to an entirely new species. He named it Masonicotuphis hamiltoni, and then for the next century, that was basically all we had. Over the decades, a few more specimens turned up, always dead, always in the stomachs of sperm whales, or tangled in the nets of fishing boats targeting Patagonian toothfish in Antarctic waters. In 2003, a New Zealand fishing vessel hauled up a specimen that was over 5 meters long and weighed more than 300 kilograms. It was preserved and put on display at the Museum of New Zealand, Te Papa, where it remains the only colossal squid on exhibit anywhere in the world. Scientists studied the dead specimens carefully and pieced together a picture of an animal that was, on paper, genuinely terrifying. The colossal squid can grow to an estimated 7 meters in length and weigh up to 500 kilograms, making it the heaviest invertebrate on the planet. Its 8 arms are lined with sharp hooks that rotate in their sockets, and its 2 longer tentacles have club-shaped tips armed with hooks that can swivel independently. Its eyes are among the largest in the animal kingdom, the size of dinner plates, evolved to detect the faintest traces of light in the deep ocean. But nobody had ever seen one alive in its natural habitat, not once in 100 years. The problem was not that we were not looking. Several expeditions had specifically set out to find and film a living colossal squid. A team called Colossal mounted 4 Antarctic missions attempting exactly that. They came close, filming what appeared to be a similar-sized glass squid during their 4th expedition in 2023, but they could not confirm the species. The colossal squid remained invisible. Part of the difficulty is the environment itself. The Southern Ocean is vast, cold, and dark. The colossal squid appears to inhabit the mesopelagic and bathypelagic zones, the twilight and midnight layers of the water column, from a few hundred meters to over 1,000 meters deep. Finding a single species, even a large one, in a 3-dimensional volume of ocean that stretches for thousands of kilometers in every direction is an almost impossible task. It is like searching for a specific bird in the entire atmosphere of Earth. But there is another reason the colossal squid has evaded us, and it is more interesting than simple geography. These animals are smart. They have large, complex brains, and they have evolved for millions of years alongside one of the most effective predators in the ocean: sperm whales. Sperm whales are the colossal squid's primary predator. They dive to extraordinary depths, using echolocation to hunt in the dark. But echolocation is not silent. A diving sperm whale displaces water, creates pressure waves, and most importantly, disturbs bioluminescent organisms as it descends. The tiny creatures that produce light in the deep ocean flash and glow when disturbed, creating a visible trail that an alert squid can detect from a considerable distance. Over millions of years of evolutionary pressure, the colossal squid has become exceptionally good at detecting approaching threats by watching for these light signatures. Now think about what we send into the deep ocean. Remotely operated vehicles with bright lights, loud motors, and mechanical arms. Submersibles that hum and vibrate and illuminate the water like a spotlight. To a colossal squid that has evolved to detect and evade the subtle light signature of a diving whale, our exploration equipment must look like a fireworks display. No wonder they run. And then, in March 2025, during a voyage that was not even specifically looking for them, the colossal squid finally appeared on camera. The Schmidt Ocean Institute's research vessel, Falkor 2, was operating near the South Sandwich Islands in the remote South Atlantic, conducting a deep-sea exploration expedition in partnership with the Nippon Foundation Nekton Ocean Sensors. The ship's remotely operated vehicle, a robot named Sebastian, was streaming live video from the deep ocean as part of a public dive stream that anyone with an internet connection could watch. And during one of these dives, at a depth of about 600 meters, a small translucent animal drifted into the frame. It was a juvenile colossal squid, about 30 centimeters long. Its body was almost completely transparent, like a glass sculpture floating in the dark water. Its fins were so delicate they were barely visible. Its eyes, which would grow to the size of dinner plates in adulthood, shone with an iridescent quality. Its arms were fanned out gracefully from its head, and on the middle of those arms, the telltale hooks of Mesonychoteuthis hamiltoni were just visible enough for experts to confirm the identification. Dr. Kat Bolstad, a squid researcher at the Auckland University of Technology in New Zealand and one of the world's leading experts on colossal squid, was among those who verified the footage. For 100 years, she said, we have mainly encountered them as prey remains in whale and seabird stomachs and as predators of harvested toothfish. This was the first time scientists had confirmed footage of the species in its natural habitat. What struck many people about the footage was how monstrous the animal appeared. For a century, the colossal squid had been described in terms designed to terrify. The largest, the heaviest, the most fearsome invertebrate, hooks that can swivel, eyes the size of soccer balls, a deep-sea kraken, and the first time we actually saw one alive at home in the ocean, it looked delicate, elegant, beautiful even. Dr. Bolstad herself made this point. "I really love that we have seen a young colossal squid first," she said. "This animal is so beautiful. It helps us to shine a bit of a light on the animal without some of that monster frenzy hype." The colossal squid was not the only first caught on camera in that period. Just 2 months earlier, in January 2025, another expedition aboard the Falkor 2 had filmed the glacial glass squid, Galiteuthis glacialis, alive in the Southern Ocean near Antarctica for the first confirmed time. Dr. Jyotika Vimani, the Schmidt Ocean Institute's executive director, noted the significance. The first sighting of 2 different squids on back-to-back expeditions is remarkable and shows how little we have seen of the magnificent inhabitants of the Southern Ocean. And squids were far from the only category producing firsts. In 2024, researchers from the Minderoo Foundation and the University of Western Australia captured rare footage of a deep-sea hooked squid, Teningia dani, attacking an underwater camera at about 1,000 meters depth in the Pacific. The squid descended on the camera, apparently mistaking it for prey, and before striking it flashed its photophores, 2 massive, bioluminescent organs at the tips of its arms. These photophores are the largest light-producing organs known in any animal on Earth. The squid was using them as headlights, blinding flashes of light designed to stun and disorient prey before an attack. Scientists had known the photophores existed, but this was one of the first times they had ever been filmed in use. Meanwhile, at the Monterey Bay Aquarium Research Institute in California, a different kind of mystery was finally being solved. For over 20 years, Mbari scientists had occasionally spotted a strange, translucent creature during deep-sea dives in the midnight zone. It used a hood-like structure around its head to catch prey and had detachable tentacles. Its hood and tail were decorated with glowing blue-green dots of bioluminescence. Nobody could figure out what it was. It was too weird for any known category. They called it the mystery mollusk. In 2025, researchers finally identified it. The creature was a nudibranch, a type of sea slug. But not just any nudibranch. It was so different from all known nudibranch that it required the creation of an entirely new family to accommodate it. They named it Bathydavius caudactylus, and its discovery meant that the deep ocean was not just hiding new species, it was hiding new families. New branches on the tree of life that we had no idea existed. The discoveries kept coming. In the Southern Ocean, an expedition uncovered a carnivorous sponge shaped like a ball, covered in tiny hooks that trap prey. Unlike most sponges, which passively filter food particles from the water column, this species actively catches and consumes other organisms. The expedition that found it also discovered 30 additional new deep-sea species on the same voyage. In January 2025, off the coast of Vietnam, researchers described a new species of supergiant isopod, a deep-sea crustacean in the genus Bathynomus. These animals are sometimes called giant sea bugs because of their resemblance to terrestrial pillbugs, but scaled up dramatically. The new species can grow over 12 inches long and earned widespread attention for its resemblance to the helmet of Darth Vader. Off the coast of Guam, researchers retrieved monitoring structures that had been sitting on the deep sea floor since 2018. These were essentially underwater hotels, stacks of plates designed to attract and house marine organisms over time. When they pulled them up after 8 years, the plates were covered in life that no human had ever seen. A transparent goby fish whose skeleton was visible through paper-thin skin. A sea slug wearing yellow polka dots. an orange fish with bulging eyes. The team identified at least 20 new species from those structures alone, and all of these discoveries came from the Ocean Census Framework, the largest collaborative effort to accelerate the discovery of marine life ever organised. By March 2025, the programme had announced 866 new marine species, not collected over a lifetime. Described in a single concentrated push, the list included new sharks, sea butterflies, mud dragons, bamboo coral, water bears, octocoral, shrimp, crabs, reef fish, squat lobsters, pipe horses, limpets, and brittle stars. An entire zoo of organisms that had been living in the ocean the entire time, unseen and uncounted. It is common for 50% of all organisms collected on a single deep-sea expedition to be new to science. On some expeditions, the figure approaches 95%. Only about 240,000 marine species have been formally described and documented. Estimates suggest that millions more exist. The deep sea holds hundreds of thousands of undiscovered species, and it makes up 95% of the available living space on Earth. We are not filling in the last few blanks on a nearly complete map. We are still drawing the map, and every time we drop a camera or pull up a net, the map gets bigger. But every one of these discoveries comes with an uncomfortable question attached to it. If we are still finding hundreds of new species every year in a habitat we have barely explored, what happens when we send something down there that is not a camera? What happens when, instead of looking, we start taking? The deep sea has a way of making us rethink the word impossible. Consider the giant isopods. These are crustaceans, related to the pill bugs you find under rocks in your garden. On land, pill bugs are tiny, about a centimetre long. In the deep sea, their relatives grow to enormous sizes. The genus Bathynomus includes species that reach over 50 centimetres, about 20 inches in length. They look like something designed for a science fiction film, with segmented exoskeletons, compound eyes that reflect light, and a body plan that has barely changed in 160 million years. They are living fossils in the truest sense, older than most dinosaurs. The phenomenon is called deep-sea gigantism, and it has puzzled biologists for over a century. In the deep ocean, many organisms grow significantly larger than their shallow water or terrestrial relatives. Giant isopods, giant amphipods, giant squid, giant tube worms. The reasons are not fully understood, and the explanations vary depending on the organism, but they likely involve some combination of cold temperatures, slowing metabolism, low predation pressure removing size constraints, and adaptation to scarce food resources that favours larger bodies capable of storing more energy between meals. The giant isopod found off Vietnam in January 2025 was a new species in the Bathynomus genus, growing over 12 inches long. Researchers described it in a formal taxonomy paper, noting its distinctive morphological features that separated it from the roughly 20 other known Bathynomus species. The discovery made international headlines, partly because of its size and alien appearance, and partly because it came from a region that is not traditionally associated with deep-sea research. Vietnam's deep waters, like those of most developing nations, have been barely explored. The fact that a species this large and this distinctive was sitting there, undiscovered in a region where people have fished for centuries, is a reminder that the ocean's inventory is nowhere close to complete. And then there are the organisms that defy categorization entirely. In deep waters off the coast of California, researchers with the Monterey Bay Aquarium Research Institute have spent decades cataloguing the life of the Midnight Zone, the region between 1,000 and 4,000 meters where no sunlight penetrates. Using remotely operated vehicles equipped with high-definition cameras, they have amassed a visual library of deep-sea organisms that is unmatched anywhere in the world. One of their most persistent mysteries was a small, gelatinous creature that kept appearing on their cameras during dives. It was translucent there, roughly the size of a fist, and it had a bizarre anatomy that did not match any known animal group. It seemed to have a hood-like structure around its head, which it used to trap food, and it had tentacles that it could apparently detach from its body and leave behind, possibly as a decoy for predators. Its body was dotted with tiny blue-green points of bioluminescence. For more than 20 years, scientists at MBARI filmed it, discussed it, and could not figure out what it was. They called it the mystery mollusk, tentatively placing it somewhere in the molluscan phylum, but with no certainty. In 2025, after years of painstaking analysis of specimens and DNA, the mystery was finally solved. The creature was a nudibranch, a sea slug. But it was so different from every other nudibranch ever described that it required the creation of an entirely new family. They named it Bathydavius caudactylus, from the Greek for deep sea and the Latin for tail finger, a reference to its extraordinary detachable tentacles. The discovery of a new family is a much bigger deal than the discovery of a new species. Families are high-level branches on the tree of life. Finding a new one is like discovering a new room in a building you thought you had completely mapped. This is not an isolated case. Deep-sea expeditions routinely find organisms that do not fit into existing categories. The carnivorous sponge discovered in the Southern Ocean in 2025 is another example. Sponges are among the simplest multicellular animals on Earth. They are typically sessile, meaning they stay in one place, and they feed by passively filtering water through their bodies, straining out tiny particles of food. The idea that a sponge could be a predator, actively catching prey, was once considered absurd. But the deep sea has produced multiple examples of carnivorous sponges, species that have abandoned the filter-feeding lifestyle and evolved tiny hooks or spicules on their surfaces that snag passing organisms. The deathball sponge found in the Southern Ocean is spherical, covered in hooks, and catches small animals that brush against it. It looks more like a medieval weapon than anything you would associate with the word sponge. The reason the deep sea produces so many surprises is partly that it is vast and poorly explored, but also that it offers ecological niches that simply do not exist anywhere else. The abyssal plains, the hydrothermal vents, the cold seeps, the trenches, the seamounts, each of these habitats has its own set of conditions, its own selection pressures, and its own evolutionary trajectory. A seamount in the mid-Atlantic is as different from a trench in the Pacific as a desert is from a rainforest, and within each of these habitats the isolation is extreme. Deep-sea currents are slow. Many organisms have limited dispersal ability. Populations can evolve independently for millions of years, producing species found nowhere else on Earth. This is what makes the deep sea so vulnerable to disturbance. When you destroy a patch of abyssal plain covered in polymetallic nodules, you are not just removing rocks. You are potentially eliminating species that exist only on that patch, species that took millions of years to evolve and that cannot be found on any other patch of seafloor anywhere in the world. The extinction risk is real, and it is amplified by the fact that we do not know what most of those species are. There is a concept in conservation biology called the extinction debt. It refers to the idea that habitat destruction causes extinctions not immediately, but with a time lag. Species lose their habitat today and go extinct decades or centuries later, as their populations slowly dwindle below viable levels. In the deep sea, this time lag could be enormous, because deep sea organisms live slow lives. Some deep sea corals are centuries old. Some clams at cold seeps can live for over 500 years. If you destroy their habitat, the last individuals might persist for generations before finally dying out. We would not even know they were gone. The slow-motion crisis is unfolding right now, in real time, and most of the world has no idea. The deep ocean does not have the visual charisma of a coral reef or a tropical rainforest. It does not have flagship species that appear on the covers of magazines. Nobody is wearing a t-shirt with a picture of a polymetallic nodule, but the ecosystems that depend on those nodules, the ecosystems that produce dark oxygen, that support unknown hundreds of endemic species, that may play a role in global nutrient cycling, Those ecosystems are no less important for being invisible to us. Science fiction taught us to think of the deep sea as a place of monsters. The reality is more interesting and more fragile. The deep sea is a place of quiet, slow, intricate life, unfolding in total darkness over geological timescales. It is a library of evolutionary experiments that has been running for billions of years, producing solutions to biological problems that we are only now beginning to read, and we are on the verge of destroying parts of it before we even open the first page. The question is whether we are paying attention. The discoveries of the last few years suggest that at least some people are. The scientists aboard the Falkor, the taxonomists at the Senckenberg Institute, the researchers at MBARI, the teams lowering cameras into Hadal trenches, they are doing the work. They are cataloguing, describing, photographing, and fighting for attention in a world that is much more interested in the latest AI model or the next space launch. They are doing this work because they understand something that the rest of us are still learning. The deep ocean is not empty. It is not irrelevant. It is not a void to be mined or ignored. It is the largest and oldest living system on this planet, and it has been here longer than we have. Longer than trees, longer than flowers, longer than insects. The deep ocean's ecosystems predate every land-based ecosystem on Earth, and they may have given rise to all of them. If life started at hydrothermal vents, then every organism on the surface, every bird, every tree, every human being is a descendant of something that once lived in total darkness at the bottom of the sea. We are not separate from the deep ocean. We are a product of it, and what we do to it, we do to our own history. The Clarion-Clipperton Zone stretches across nearly 4,500 miles of the Pacific Ocean floor from the coast of Mexico to Hawaii. It is one of the most mineral-rich regions on Earth, and the minerals are just sitting there. Polymetallic nodules, those same potato-sized lumps of manganese, nickel, cobalt, copper, and lithium that might be producing dark oxygen, are scattered across the abyssal plain in quantities that the mining industry describes as staggering. By some estimates, the CCZ alone contains enough critical metals to meet global demand for electric vehicle batteries for decades. 16 companies currently hold exploration contracts in the CCZ, granted by the International Seabed Authority, a United Nations body responsible for managing the ocean floor beyond national borders. The legal framework that governs this region is the United Nations Convention on the Law of the Sea, which designates the international seabed as the common heritage of mankind. That phrase sounds protective, and it was meant to be, but it also left the door open. Countries can vote to open the seabed to commercial mining if they agree on a set of rules, a mining code. And the negotiations over that code have been grinding along for years, with industry lobbying for access and conservation groups arguing for a moratorium until we understand what we would be destroying. In December 2025, the most comprehensive assessment of deep-sea mining impacts yet conducted was published in Nature Ecology and Evolution. A research team led by Eva Stewart studied the aftermath of an industrial-scale mining trial in the CCZ, in which a collector vehicle physically scraped the seafloor, vacuuming up nodules in the sediment around them. The researchers mapped the biological communities before and after the trial, cataloguing every organism they could find. The results were sobering. The team identified 788 deep-sea species in the study area, many of which had never been described before. In the tracks left by the mining machine, animal abundance dropped by 37% and species diversity dropped by 32%. A third of the species gone from the mined tracks, and these were not species that would bounce back in a season or even a century. Polymetallic nodules form at a rate of a few millimeters per million years. Once they are scraped off the seafloor, they do not regenerate on any time scale that matters to human civilization. The habitat is not damaged, it is erased. This matters because the deep-sea floor is not the barren wasteland that our lingering Forbes-era assumptions tell us it is. The organisms that live on and around polymetallic nodules are adapted to those specific conditions. Some of them are obligate, meaning they live only on or near nodules and cannot survive without them. Remove the nodules and you remove the foundation of their ecosystem. It is not like logging a forest that can regrow. It is like paving over a meadow with concrete. The wildflowers are not coming back. A 2020 paper published in Trends in Ecology and Evolution, authored by a large team of deep-sea scientists from institutions around the world, laid out the problem in stark terms. Scientific misconceptions about the deep sea, they argued, were leading to systematic underestimation of the environmental impacts of seabed mining. People assume the deep sea is resilient because it seems vast and empty. In reality, deep-sea ecosystems are fragile, slow to recover, and far more biodiverse than casual observers imagine. Deep-sea corals on seamounts can live for centuries. Growth rates of many deep-sea organisms are measured in millimeters per year, if that. Reproductive cycles are slow. Larval dispersal is poorly understood. We do not know how connected these communities are to one another, or what happens to the broader ecosystem when one node in the network is destroyed. The paper identified a specific set of misconceptions that were driving flawed decision-making. People assumed the mining footprint was small, relative to the total area of the CCZ. In reality, when you account for the fact that mining targets specific habitats, the proportion of a given habitat type that would be destroyed is much larger than the proportion of the total seafloor affected. Mining companies talk about percentages of the total CCZ. Ecologists talk about percentages of the specific nodule-rich habitat type that supports the affected species. The numbers look very different depending on which denominator you use, and the physical impact extends far beyond the tracks of the mining vehicle itself. When a collector scrapes the seafloor, it kicks up enormous plumes of sediment that can drift for tens or hundreds of kilometers, settling on organisms and habitats far from the mining site. These sediment plumes can smother filter feeders, clog the respiratory structures of animals, and alter the chemistry of the water column. In a region where the natural rate of sediment disturbance is essentially zero, introducing clouds of particulate matter is an ecological event that has no precedent. This is the paradox of deep-sea mining. The very thing that makes the CCZ attractive to miners, its vast deposits of critical metals, is also the thing that makes its ecosystem unique and potentially irreplaceable. The nodules are not just rocks. They are habitat. They may be oxygen producers. They are the foundation of a biological community we barely understand. But mining is not the only threat. While the political debate over nodule extraction plays out in conference rooms in Jamaica, a slower and less visible force is already reaching the deep ocean. Climate change. For a long time, researchers assumed that the deep ocean was largely insulated from the effects of surface warming. The logic seemed sound. The deep sea is separated from the surface by thousands of meters of water. Temperatures at the bottom are already near freezing and change very slowly. The global ocean circulation that delivers surface water to the deep operates on a timescale of centuries. How could a few degrees of warming at the surface possibly affect an ecosystem 5 kilometers down? In March 2026, a study published in the Proceedings of the National Academy of Sciences by researchers at the University of Illinois Urbana-Champaign offered a surprising answer. The team studied Nitrosopumilus maritimus, an archaeon, a type of single-celled organism that belongs to a domain of life, distinct from both bacteria and the familiar multicellular organisms we usually think about. This species and its close relatives are not obscure microbes. They make up an estimated 30% of all microbial plankton in the ocean. They are everywhere, from the sunlit surface to the deepest waters, and they play a critical role in the nitrogen cycle, converting ammonia into nitrate. This process influences plankton growth, which in turn affects the entire ocean food web, including the rain of organic matter that feeds deep-sea ecosystems. The researchers found that Nitrosopumulus and its relatives were far more responsive to temperature changes than previously believed. When exposed to warmer conditions, these microbes altered their metabolism in unexpected ways, changing how they processed iron and nitrogen. The implications were alarming. Ocean warming effects may extend to depths of 3,280 feet or more, said Wei Qin, a microbiology professor at the University of Illinois and lead author of the study. If climate change is affecting the metabolism of organisms that comprise nearly a third of all microbial plankton, then the effects ripple through the entire deep-sea ecosystem. Plankton growth rates change, the quantity and quality of marine snow, the rain of organic particles that feeds the deep changes. The organisms that depend on that rain, from amphipods to snailfish, are affected. The deep ocean, it turns out, is not insulated from the surface world at all. It is connected intimately and inescapably. This creates a double threat that is difficult to overstate. On one hand, the mining industry is preparing to physically remove habitat from the deep-sea floor, destroying nodule fields that took millions of years to form and that support communities of species we have not yet catalogued. On the other hand, climate change is altering the fundamental chemistry and biology of the deep ocean from the top down, affecting the food supply, the temperature regime, and the metabolic processes of organisms that underpin the entire ecosystem, and we are proposing to do both of these things simultaneously, to an environment that we have explored less than 1,000th of 1% of. The deep-sea mining debate often gets framed as a trade-off. We need cobalt for batteries, we need nickel for electric vehicles. The green energy transition depends on metals that are currently mined on land in ways that are also environmentally destructive. Mining the ocean floor, the argument goes, might be the lesser evil, but that argument depends on an assumption that we know what we would be losing, and the honest answer is that we do not. We cannot make an informed trade-off when one side of the equation is almost entirely unknown. We are being asked to weigh the economic value of deep-sea minerals against the ecological value of deep-sea ecosystems, and we have barely begun to calculate the second number. Consider what we have already found in the CCZ alone: hundreds of new species, potential dark oxygen production by polymetallic nodules, complex microbial communities that influence global nutrient cycles, and all of this from a tiny fraction of the region that has actually been surveyed. There is a line in the deep-sea science community that captures this dilemma perfectly. We are writing the mining regulations for the deep ocean before we have finished reading the table of contents of the deep ocean's biology book. We do not know how many pages there are. We do not know what most of them say, and we are already talking about tearing some of them out. But maybe the most unsettling part of this story is not what we stand to lose in the economic sense. It is what the deep ocean might be telling us about something far bigger. Because the discoveries we have been talking about, the vents, the dark oxygen, the chemosynthetic ecosystems thriving in total darkness, they do not just challenge our understanding of the deep sea, they challenge our understanding of where life itself came from. And the answer may be stranger and more profound than anyone expected. Before 1977, the story of life's origin was a surface story. It started with Charles Darwin's famous Warm Little Pond, a shallow pool on primordial Earth where simple chemicals, bathed in sunlight and stirred by lightning, somehow assembled themselves into the first living cell. The idea evolved over the decades, refined by experiments like Stanley Miller and Harold Urey's famous 1953 experiment, in which they zapped a mixture of gases with electrical sparks and produced amino acids, the building blocks of proteins. The message was clear. Life began at the surface in the light, in the warmth. The sun was the engine. The sky was the stage. But the warm little pond had problems. Early Earth was not a gentle place. 4 billion years ago, the planet was being bombarded by asteroids and comets at a rate that would have sterilized any surface body of water multiple times over. The atmosphere was toxic, loaded with methane, ammonia, and carbon dioxide. Ultraviolet radiation from the sun, unfiltered by the ozone layer that did not yet exist, would have shredded most organic molecules as fast as they formed. Whatever life was trying to get started on the surface was facing a gauntlet of destruction. The deep ocean was different. Thousands of meters below the surface, the early deep sea was shielded from asteroid impacts, protected from UV radiation, insulated from the chaos above. And at hydrothermal vents, the conditions were intriguing. You had heat. You had mineral-rich water full of iron, sulfur, nickel, and other elements that can catalyze chemical reactions. You had chemical gradients, the difference in chemistry between the hot vent fluid and the cold surrounding seawater, which is exactly the kind of disequilibrium that drives chemical work. And you had an enclosed environment, porous rock and mineral chimneys that could concentrate reactants and protect fragile organic molecules from being diluted into the vast ocean. When the hydrothermal vent ecosystems were discovered in 1977, and when chemosynthesis was confirmed as the energy source powering those ecosystems in 1981, a new hypothesis for the origin of life began to take shape. What if life did not start in a warm little pond under the sun? What if it started in a hot little crack at the bottom of the ocean in total darkness, powered by the chemistry of the Earth itself? The idea gained traction through the 1980s and 1990s. A German chemist and patent lawyer named Gunther Wechtershäuser proposed that early life could have emerged on the surfaces of mineral crystals, particularly iron sulfide, which forms naturally at hydrothermal vents. The mineral surfaces could have acted as catalysts, promoting the formation of organic molecules from simple precursors like carbon dioxide and hydrogen sulfide. No sunlight required, no lightning needed, just hot rock, chemical gradients, and time. The hypothesis was refined further by researchers like Mike Russell at the NASA Jet Propulsion Laboratory, who proposed that a specific type of hydrothermal vent, alkaline vents, provided conditions that were particularly favourable for the origin of life. Alkaline vents are different from the dramatic black smokers that spew 400-degree fluid. They are cooler, gentler, and produce porous chimney-like mineral structures that create natural compartments, tiny chambers where chemical reactions could be concentrated and sustained. These natural compartments, Russell argued, function like primitive cells, concentrating reactants and creating the chemical conditions necessary for the emergence of metabolism. The most famous example of an alkaline vent system is the Lost City hydrothermal field, discovered in the year 2000 on the Mid-Atlantic Ridge. Unlike black smokers, which are driven by volcanic activity and tend to be short-lived, Lost City is powered by a chemical reaction between seawater and the rock of the ocean crust, a process called serpentinization. This reaction produces hydrogen, methane, and heat, and it can sustain a vent system for tens of thousands of years, far longer than the few decades typical of a black smoker. Lost City's chimneys are made of calcium carbonate, towering white columns rising from the seafloor, and they host thriving microbial communities that run entirely on chemosynthesis. The chemistry at Lost City and similar alkaline vent systems bears a striking resemblance to the chemistry inside modern living cells. The energy-producing processes in our mitochondria, the organelles that power every cell in your body, involve pumping protons across a membrane to generate a chemical gradient, which then drives the synthesis of ATP, the energy currency of life. At alkaline vents, a similar proton gradient exists naturally, produced by the difference in chemistry between the alkaline vent fluid and the slightly acidic surrounding seawater. The parallel is so close that some researchers believe the earliest forms of life essentially co-opted the natural electrochemistry of the vent environment, borrowing a trick from geology before biology even properly existed. The evidence is circumstantial but compelling. The deepest branches of the tree of life, the organisms that genetic analysis suggests are closest to the common ancestor of all living things, are thermophilic. They love heat. They are anaerobic. They do not use oxygen, and many of them are chemosynthetic, deriving their energy from hydrogen, sulfur, and other chemicals rather than sunlight. These organisms look like creatures that evolved at a hydrothermal vent. They have the metabolic fingerprint of the deep. Chemosynthesis itself appears to be ancient, far older than photosynthesis. The earliest forms of chemosynthesis did not require oxygen. They ran on hydrogen sulfide, methane, and ammonia, all of which are abundant at hydrothermal vents. Oxygen-using metabolism, the kind that powers all animal life, including ours, came much later, only after photosynthetic cyanobacteria had been pumping oxygen into the atmosphere for hundreds of millions of years. The timeline suggests that life started in the dark, running on chemistry, and only later moved into the light, and then came the dark oxygen discovery, adding yet another twist. If polymetallic nodules on the seafloor can generate oxygen through electrochemistry, then it is at least conceivable that similar mineral formations on the early Earth could have done the same thing. The rocks of the early ocean floor were rich in metals. The water was full of dissolved minerals. The conditions for electrochemical oxygen production may have existed long before any photosynthetic organism evolved. This is speculative, and the researchers are careful to frame it as a possibility rather than a conclusion. But the implication is extraordinary. The deep ocean may have had small, localized sources of oxygen before the Great Oxidation Event, the moment roughly 2.4 billion years ago, when photosynthetic organisms finally pumped enough oxygen into the atmosphere to fundamentally change the chemistry of the planet. Jeff Marlow, the Boston University biologist who co-authored the dark oxygen study, spelled out the implications clearly. The fact that oxygen can be made independently of the surface world has a lot of exciting implications for the distribution of animal life, not only in Earth's oceans, but also potentially beyond our planet. Oxygen provides enough energy for big organisms like animals to survive, so a process that makes oxygen without sunlight could allow for larger life forms to inhabit the deep, dark waters of ocean worlds like Enceladus or Europa. And this is where the story connects back to the moons of Jupiter and Saturn in a way that the hydrothermal vent discovery first hinted at nearly 50 years ago. Europa, one of Jupiter's 4 large Galilean moons, has a global ocean of liquid water beneath a shell of ice that is roughly 15 to 25 kilometers thick. The ocean is estimated to be about 100 kilometers deep, containing roughly twice as much water as all of Earth's oceans combined. Tidal heating from Jupiter's immense gravity keeps the water liquid, and there is strong indirect evidence that hydrothermal vents may exist on Europa's seafloor, driven by the same kind of tidal flexing that heats the interior. Enceladus, a tiny moon of Saturn only about 500 kilometers in diameter, punches far above its weight in terms of astrobiological interest. The Cassini spacecraft discovered geysers erupting from its south pole in 2005, plumes of water vapor and ice particles shooting hundreds of kilometers into space. Cassini flew through those plumes and analyzed their composition. It found salt. It found organic molecules. It found silica nanoparticles, which on Earth are associated with hydrothermal activity at temperatures above 90 degrees Celsius. And it found molecular hydrogen, a chemical signature that is consistent with active serpentinization, the same process that powers the Lost City hydrothermal field on Earth. If life started at hydrothermal vents on Earth, then Europa and Enceladus have the ingredients. They have liquid water, they have chemical energy, they have mineral-rich seafloors, they may have hydrothermal vents, and if dark oxygen production is a real phenomenon, they might even have a source of oxygen independent of sunlight. Before the deep-sea discoveries of the last 50 years, the search for life beyond Earth was essentially a search for another Earth. A rocky planet, the right distance from its star, with an atmosphere and liquid water on the surface. The hydrothermal vent discovery expanded that search to include any world with an ocean and a heat source. The dark oxygen discovery, if confirmed, expands it even further to any world where the right minerals interact with the right chemistry, regardless of whether a star is shining on it. We went looking for strange creatures in the deep ocean. We found evidence that the deep ocean might be where all creatures originally came from, not just the weird ones that live at vents and seeps and whale falls. All of us. Every organism that breathes, that eats, that moves, that thinks. The deep ocean may be the cradle, the original factory where the chemistry of life was first assembled, in darkness, under pressure, far from the sun. And we nearly started mining it before we figured that out. The deep sea does not volunteer its secrets easily. It gives them up one expedition at a time, one camera drop at a time, one stubborn researcher at a time, and every secret it reveals makes the previous picture look incomplete. We thought it was dead. It was alive. We thought it needed sunlight. It did not. We thought we understood its limits. We were wrong. We thought oxygen needed photosynthesis. Maybe it does not. We thought life started at the surface. Maybe it started at the bottom. And the terrifying, wonderful, humbling truth is that we have barely started looking. Think about what that means for a moment. On Enceladus, a moon so small you could drive around it in a day, there is a global ocean of liquid water that is doing chemistry. Salt, organic molecules, silica nanoparticles that on Earth are only produced at temperatures above 90 degrees Celsius, temperatures associated with hydrothermal activity, molecular hydrogen, which is the signature of serpentinization, the same geological process that powers the Lost City hydrothermal field at the bottom of the Atlantic. Every single ingredient that chemosynthetic life needs on Earth has been detected on Enceladus. Every one. Europa is even more tantalizing in some ways, because its ocean is vastly larger. It contains roughly twice as much water as all of Earth's oceans combined, locked beneath an ice shell that is between 15 and 25 kilometers thick. The Galileo spacecraft detected magnetic field anomalies consistent with a salty, conductive ocean. More recently, the Hubble Space Telescope observed what appeared to be plumes of water vapor erupting from Europa's surface, similar to the geysers on Enceladus, though these observations have been harder to confirm. NASA's Europa Clipper mission, launched in 2024, is on its way to conduct detailed flybys beginning in 2030, equipped with instruments designed to analyze the composition of Europa's surface ice and any plumes it encounters. If life exists on Europa or Enceladus, it almost certainly does not look like life on Earth's surface. There would be no plants, no trees, no animals that photosynthesize. But it might look very much like life at Earth's hydrothermal vents, microbial communities running on hydrogen and sulfur, perhaps simple multicellular organisms feeding on those microbes. Perhaps in Europa's vast ocean, something more complex, something that has had billions of years to evolve in isolation, in an environment remarkably similar to the one where life on Earth may have started. The deep ocean taught us that life does not need the sun. It needs energy, water, and chemistry. Those three things exist on at least 2 moons in our own solar system. The deep ocean did not just change how we understand our own planet, it changed how we understand the universe of places where life might be hiding, and the list of those places keeps growing. Every time we recalibrate our expectations about what life can tolerate, what it can use as fuel, what conditions it can survive, we expand the zone of possibility. The hydrothermal vents expanded it. The chemosynthetic ecosystems expanded it. The snailfish at 8,336 meters expanded it. The dark oxygen expanded it. Each discovery is not just a fact about a specific organism or a specific chemical reaction. Each discovery is a revision of the boundary between possible and impossible, a boundary that life keeps pushing outward while we scramble to keep up. And the source of all of these revisions, every single one, is the same place. The deep ocean, the place we declared dead 180 years ago, the place we have explored less of than the surface of Mars, the place that, despite our neglect and our ignorance and our persistent underestimation, keeps delivering discoveries that reshape our understanding of what it means to be alive. Let us sit with the numbers for a moment, because the numbers are the most honest thing in this entire story. The deep ocean, defined as everything below 200 meters, covers 66% of the entire planet. Not 66% of the ocean, 66% of Earth. It is the single largest ecosystem in existence, and it is the least explored place, accessible to human technology. We have sent robots to Mars, landed probes on comets, and photographed black holes, but we have visually observed less than 1,000th of 1% of our own ocean floor. That number comes from a study published in Science Advances in 2024, and it is worth repeating because it sounds too small to be true. The total area of deep sea floor that has ever been seen by human eyes, either directly through a submersible window or through the camera of a remotely operated vehicle, adds up to roughly 1/10 the size of Belgium. That is the sum total of all deep-sea visual exploration in human history. Every dive, every ROV survey, every submersible descent since we first started looking, all of it combined amounts to a postage stamp on a basketball. And the distribution of that exploration is wildly uneven. 65% of all the deep-sea observations in the dataset were made within 200 nautical miles of just 3 countries: the United States, Japan, and New Zealand. If you live anywhere else on Earth, the deep ocean off your coast is almost completely unseen. 97% of all deep-sea dives ever compiled were conducted by just 5 countries. The remaining nations of the world have contributed almost nothing to the visual record of the largest habitat on the planet. This is not because the deep ocean is unimportant. It is because it is expensive. A single deep-sea research expedition can cost millions of dollars. Submersibles and remotely operated vehicles are complex machines that require specialized ships, trained operators, and extensive support infrastructure. Governments, facing competing demands on their budgets, have consistently chosen to invest elsewhere. On average, countries spend about 1.7% of their annual research budgets on exploring beneath the ocean surface. More people have travelled to space than have visited the deep ocean floor. We have more detailed maps of the surface of Mars than we do of the bottom of the Pacific. The irony is almost too neat. We scan the skies for signs of extraterrestrial life, while ignoring the alien world beneath our own feet. A world that, unlike Mars, is confirmed to be teeming with life we have not yet catalogued. The Ocean Census, launched in 2023 as the largest collaborative effort to accelerate the discovery of marine life, announced 866 new species in its first major reporting milestone in March 2025. Those species included new sharks, sea butterflies, mud dragons, bamboo coral, water bears, octocoral, sponges, shrimp, crabs, reef fish, pipe horses, limpets, and brittle stars. And they were the product of a deliberately accelerated process, one designed to shorten the agonizing gap between collecting a specimen and formally describing it. That gap is itself a revealing detail. There are thousands of specimens sitting in museum collections and research labs around the world, collected on deep-sea expeditions years or even decades ago, that have never been formally described and named. The bottleneck is not discovery, it is taxonomy. We are finding new species faster than we can process them. The ocean delivers, and our bureaucratic and scientific systems cannot keep up. Only about 240,000 marine species have been formally documented and given scientific names. Current estimates suggest that the total number of marine species, including those not yet described, may be between 1 and 2 million. Some estimates go higher. We have catalogued somewhere between 10 and 25% of the life in our own ocean. The rest is waiting, unnamed and unstudied, in water we have not visited yet. And this is not a problem that will fix itself. Deep-sea species face the same threats as the rest of the natural world: habitat destruction, pollution, climate change, and now the looming prospect of industrial mining, but with an additional: cruelty. Most of them will never be known. They will go extinct before a taxonomist ever gets a chance to describe them. We are losing species we have not yet discovered. The murder weapon is being used before the body is found. This is the central tension of deep-sea science in the 21st century. We know enough to be concerned, but not enough to be confident. We know the deep ocean is biodiverse, fragile and connected to the surface world in ways we are just beginning to understand. We know that human activities, from mining to carbon emissions, are reaching deeper than we assumed. But we do not have a complete inventory of what is down there, how it is connected, what it does, or what will happen if we break it. The discoveries of the last few years have done something important. They have given us a taste of how much we are missing. The colossal squid, filmed alive for the first time in a century of searching. Dark oxygen produced by rocks in total darkness. Fish surviving at depths their own biology should not allow. Entire families of organisms that we did not know existed until this year. These are not marginal findings at the edge of an otherwise well-mapped territory. These are fundamental surprises coming from a place we thought we understood, and every surprise has followed the same pattern. We make an assumption based on the tiny fraction of the deep ocean we have explored. We treat that assumption as settled science, and then the deep ocean proves us wrong. We assumed nothing lived below 300 fathoms. The HMS Challenger proved us wrong in the 1870s. We assumed all life depended on sunlight. Hydrothermal vents proved us wrong in 1977. We assumed the deep ocean was a biological desert. Chemosynthetic ecosystems proved us wrong in the 1980s. We assumed fish could not survive below 8,200 meters. The snailfish proved us wrong in the 2020s. We assumed oxygen could only be made by photosynthesis. Polymetallic nodules may be proving us wrong right now. There is a pattern in these corrections, and it is worth naming explicitly. Every time we have been wrong about the deep ocean, we have been wrong in the same direction. We have underestimated it. We have assumed it was less complex, less alive, less important, and less capable than it actually is. Not once in the history of deep-sea exploration has a discovery revealed that the ocean was emptier or simpler than we thought. Every single time the answer has been, there is more going on down here than you imagined. This is not because scientists are careless. It is because the deep ocean is genuinely difficult to study. You cannot walk through it. You cannot set up a permanent camp. Every visit requires millions of dollars of specialized equipment, and you can stay for only a few hours before the submersible has to ascend. Studying the deep sea is like trying to understand a continent by looking at it and through a keyhole for 5 minutes at a time. But there is also a psychological dimension. Humans are surface creatures. We evolved on land, in the light, surrounded by things we can see and touch and hear. The deep ocean is the antithesis of our natural environment. It is dark where we need light. It is crushing where we need pressure balance. It is cold where we need warmth. It is silent where we expect sound. Our instincts, honed over millions of years of terrestrial evolution, tell us that places like the deep ocean are hostile, empty, and dead. Our instincts are wrong. There is an academic paper published in 2021 in the ICES Journal of Marine Science by a researcher named Alan Jameson, who is both one of the world's leading deep-sea scientists and one of the most vocal critics of how the deep ocean is portrayed in popular media. The paper is titled Fear and Loathing of the Deep Ocean, and it argues that cultural biases, rooted in ancient associations between depth, darkness, and the underworld, have consistently led the public and even some scientists to underestimate the deep sea. We describe the Mariana Trench by comparing it to Mount Everest turned upside down. We name its zones after Greek gods of death. We call its inhabitants monsters and nightmares. And then we wonder why nobody cares about protecting it. Jameson points out that no other habitat on Earth is consistently introduced to the public through the lens of what we do not know about it, rather than what we do. Every documentary about the deep sea emphasizes mystery, danger, and the unknown. Nobody makes a documentary about forest that spends half its runtime talking about how scary and unexplored forests are. But deep-sea documentaries routinely frame the entire subject as a venture into the terrifying void. This framing serves entertainment, but it undermines conservation. You cannot motivate people to protect something they have been taught to fear. The truth is simpler and more beautiful than the fear narratives suggest. The deep ocean is alive. It is complicated. It is full of organisms that are elegant, fragile, ingenious, and strange. It supports ecosystems that run on principles we only recently discovered. It may hold the answer to where life on Earth began. It almost certainly holds the key to understanding where life might exist elsewhere in the solar system. And it is far more vulnerable than it looks. We live on a blue planet. Water covers 71% of the surface, and the deep ocean makes up the vast majority of that water. We call ourselves an advanced technological civilization, and yet we have seen less of our own planet's floor than we have of the surface of the moon. We send billionaires to the edge of space for fun, while the deepest ocean trenches remain among the least visited places in existence. Only 27 people in all of human history have descended to the bottom of the Challenger Deep. About 600 have been to space. The question that the deep ocean keeps asking us, the question that it has been asking since Edward Forbes declared it dead in the 1840s and the HMS Challenger proved him wrong 30 years later, is this: what else are you missing? Because the answer, based on everything we have found so far, is almost everything. We have been wrong about the deep ocean for 200 years. We have been wrong about what lives there, wrong about what survives there, wrong about what is possible there, and wrong about what it means for us. Every decade brings a discovery that reshapes our understanding, and every discovery makes the previous decade's assumptions look naive. The cold spot in the cosmic microwave background, the early galaxies found by the James Webb Space Telescope, the Hubble tension in cosmology. These are the kinds of anomalies that signal a paradigm shift, a moment when the old model starts to crack under the weight of what it cannot explain. The deep ocean has its own version of this: the hydrothermal vents, the dark oxygen, the impossible fish, the monsters that turned out to be beautiful, the ecosystems we are destroying before we have finished discovering them. Each one is a crack in the old assumption that the deep ocean is a void, a gap, an absence. It is not an absence. It is a presence we have barely begun to understand. The deep sea makes up 95% of the available living space on Earth. It holds hundreds of thousands of undiscovered species. It runs on chemistry that we only identified in the last 50 years. It produces oxygen through processes we only discovered last year. It harbours creatures we have been searching for a century and are only now finding. And it may be the birthplace of all life on this planet. We are not at the end of deep-sea discovery. We are not even in the middle. We are at the very beginning, standing on the edge of the largest, darkest, most mysterious habitat on our planet, having seen almost none of it, and already overwhelmed by what the tiny fraction we have explored has revealed. The next time you look at the ocean, remember that what you see on the surface, the waves, the sunlight, the blue, that is the skin. Beneath it, stretching down for miles, is a world that is older, stranger, and more alive than anything we have on land. A world that operates by rules we are still learning. A world that has been there for billions of years, running perfectly well without our knowledge or our permission. We were wrong about the deep sea. We have been wrong about it for a very long time, and the wonderful, terrifying, humbling truth is that we are probably still wrong about most of it, because the ocean is under no obligation to match our expectations, and every time we thought we understood it, it showed us something new. That is not a failure, that is the whole point. A universe that makes sense is a small universe. An ocean that matches our predictions is a shallow ocean. The deep, the truly deep, is always stranger than we imagine. And the fact that we keep being surprised is not a sign that we are doing science badly. It is a sign that we are doing it at all, that we are still looking, that we are still sending cameras into the dark, still pulling creatures out of the abyss, still asking what else is down there. And the answer so far has always been the same: more than you think, more than you dreamed, more than you will ever finish counting. Sleep well knowing that you live on a planet with a heartbeat you have never heard. Miles below the waves, in water so heavy it would crush a submarine, there are animals hunting, bacteria farming, rocks making oxygen, and entire ecosystems humming along in total darkness. They do not know about you. They have never seen the sun, and they have been there for longer than anything else alive. The deep ocean is the oldest, biggest, strangest room in the house, and we just found the door. If this kept you company tonight, or if it changed the way you think about the water covering most of this planet, the subscribe button is right there. I make videos like this one, the kind that sit with you for a while after they are over, every week. And if you want to go even deeper, there is a video on screen right now that will take you into another corner of the unknown. Thanks for being here. I will see you in the next one.